Good afternoon. I'm Baharudin Ibrahim. I'm a clinical lecturer in School of Pharmaceutical Sciences, University of Science Malaysia. So today I'm going to talk about this personalized medicine. So I'm just going to give a brief introduction on how to use this concept of genomics, proteomics and metabolomics in developing new drug therapy based on this personalized medicine. So I'll be very brief and try to be as informative as possible, so at least I can convey the message that I'm planning to convey today, so at least you have some understanding. So today I'll be talking about personalized medicine. So as I said, how to use genomics and pharmacogenomics, proteomics and pharmacoproteomics, metabolomics and pharmacometabolomics in applying this personalized medicine. And lastly, I'll be talking about these challenges in personalized medicine. So what's the current treatment approach? So it's based on large cohort studies. So if you see, even though the drugs are developed based on molecules, they look at the pathophysiology and develop the drugs, but at the end when they do a clinical trials, they always go for the disease. So it's always based on one size fits all model. So they expect that, for example, a hypertension patient or diabetic patients, they all have a similar effect to the drugs. So it's more on disease oriented, for example anti-hypertension and anti-diabetic drugs. So you don't phenotype or stratify the patients based on their molecular information, and that means they do not account for individual differences. And this also limited to patient signs and symptoms, medical and family history and investigational tests in tailoring individual care, and it's more on a reactive approach. So once you have the disease, then only you can given the treatment. So this caused a lot of drug development failure in large clinical trials. So for the drug development failure, so as been talked by a few other speakers before this, so drug development is a very costly and time-consuming process. So it can probably cost about 100 to 100 million US dollars and probably 10 to 20 years of time. And even one particular companies, they may just depend on that particular drugs, and if that fails, maybe they can just go bankrupt. And drugs with small benefits need larger sample size for significant results. So if you see some drugs, they only have a very small effect size. So when you do a clinical trial, sometimes if you do in a small sample size, you may not see the significant difference between the trial drugs and the placebo, and that can cause also the drug failure. And a lot of statistics have shown that 85% therapy fail in early clinical trials and another half in phase 3 trials. So a lot of drugs just go inside the dustbin without being for further trials. So the failures of this could be multiple factors. So as also pointed out by, I think, Dr. Prakash just now. So the main, I think Dr. Jayaprakash did mention about efficacy, safety and the cost. And these issues actually cause the failure of the drug. So the safety, the efficacy, inappropriate study design, and also they forgot about this patient heterogeneity. So we all must remember that every patient or every individual is unique, and everyone has different response to a drug. So even if you see a simple antihistamines, if some of you may have effect taking just a simple chlorpheniramine, Piriton, and some of you feel that it doesn't affect or give a good effect to you guys, and you may take loratadine or cetirizine or different things. So you can see actually different drugs have different effects on different individuals, and that's what we need to take into consideration for future drug development. So these are some of the examples of drug failures. So for example, dimebon in Alzheimer, ocrelizumab in rheumatoid arthritis, taspoglutide in diabetes, and tifacogen for sepsis or severe community acquired pneumonia. So for example, this tifacogen, initially it was developed to treat general sepsis. So they did a large clinical trial, phase 3 trials, comparing patients with general sepsis and the placebo and the treatment drugs, and they found it actually didn't show any significant difference between the two groups. So what they did is, these investigators, they actually looked within the sample, and they found that probably those who are having community acquired pneumonia, they may have some effects on the tifacogen. So again they tried another trial, multi-center trial, and just focusing on community acquired pneumonia patient, but eventually the drug even didn't show any improvement in terms of mortality. So this is the source from Ledford, Nature. So it shows that from 1990s to 2002, the failure of drugs, especially in phase 3 clinical trials, you can see the red line, is actually increasing. So there are more and more drugs are actually failing in clinical trials. And again, the reasons they are found is mostly are, if you see the pie chart here, the blue color is mostly on the efficacy, and the probably yellow color is more on safety. So you can see most of the drugs fail due to this safety and efficacy. And so how are we going to overcome this problem? So one way of it is by using this personalized medicine concept. So what is personalized medicine? So it's actually use of patients' characteristics, including demographics, histories, and molecular information. So the key point here is the molecular information. So you use the either genetic, protein, and metabolic profile to better define therapies. So it's actually an integration of diagnostic and therapeutics known as theranostics. I think also mentioned by one of the speakers. The concept of theranostic is a diagnostic therapy that identifies patients most likely to be helped by new medication and targets drug therapy based on the test results. So actually, even though, for example, it's a breast cancer patient, for example, this drug Herceptin, you don't actually target whole breast cancer group of the patients, but actually you target certain molecules which only will be benefited by the drugs. For example, this Herceptin and Herceptest. So the philosophy behind this personalized medicine is every patient has a unique biology and pathophysiology that should be reflected in the choice of pharmacotherapy, thus resulting in an improved treatment outcome. So the key word here is every patient is unique, and the choice of pharmacotherapy should be based on that individual patient. So why is personalized medicine better than the current approach? So it treats the patient, not just disease. So for example, if you are a breast cancer patient, it doesn't just treat the disease breast cancer, but actually go for more on the individual patient, how the patient's genetic or protein or metabolic profile. So it predicts susceptibility and risk factors, preempt disease progression, target intervention, avoid ineffective treatments, and prevent adverse reactions. So these are some of the things that can be done by personalized medicine, and it reduces costs in the long term. Of course, in the beginning, when you want to try to identify all this molecular information, it will be costly. But when you do clinical trials later on, then it can actually save costs. Even when you go for the treatment for the real patient, that can also reduce costs in the long term. And it's provide for individual differences, a tailored approach. And it can also stratify genotype and phenotype diseases. So many diseases, previously it was classified as one group. For example, this asthma and COPD. Before this, I think in 1970s, they had this Dutch hypothesis where they actually clumped them together, asthma and COPD, as one disease. And then comes this British hypothesis where they separate them. But now they are even looking at the smaller part, because they found that there are some overlapping characteristics between this asthma and COPD. And they found that probably we don't treat based on just the disease, but actually we can actually phenotype them based on their neutrophil count, their eosinophils count, and so on. So we can use this personalized medicine to do that as well. And numerous factors contribute to variable drug response, including age, sex, body weight, nutrition, organ function, infections, co-medications, and genetic factors. So this is the omic structure, where usually it starts from genomics. Genomics is a study of all the genes, and then it goes down to transcriptomics, a study of mRNA, and this will be transcribed to proteins, which is proteomics, study of proteins. And the last, the functional aspect, is the metabolomics, a study of metabolites. So initially they started to look at genomics, and then they go for transcriptomics, proteomics, and now they are looking at metabolites level to identify disease markers. So as I said, genomics study of all the genes in the living organism, and this pharmacogenomics is study that examines the impact of genetic variations on the response to medications. So basically they are looking at the gene, and then they can predict how the drugs is going to respond. So even when you want to develop the new drugs, usually they will go for the pathophysiology, they will find all the things, but when they go for clinical trials, they should be also based on the same concept, based on the gene or protein profile, instead of going for the disease. And you can use this pharmacogenomic concept to target the patients. And drug metabolizing enzymes, transporters and receptors are encoded by several hundred genes, playing a pervasive role in ADME and drug targeting. So the reason we are looking at the genes is because the genes are the one responsible for how the drug is going to be responsible. It's a kind of romantic kind of things. Okay, genes is the one determine how the drug is going to be response in the body. So that's why we are studying the genes. But the limitations about this genomics concept is a major factor underlying inter-individual variation in drug effects is variation in metabolic phenotype, which is influenced not only by genotype but also by environmental factors such as nutritional status, the gut microbiota age, disease, and co- or pre-administration of other drugs which modulate drug pharmacokinetic efficacy and toxicity. So the effect of drugs they found is not just by the gene but also a lot of environmental factors or patient factors also contribute to the effect of the drug. So thus, although genetic variation is clearly important, it seems unlikely that personalized drug therapy will be enabled for a wide range of major diseases using genomic knowledge alone. So then they went to study on proteomics, which is comprehensive analysis and characterization of all the proteins and protein isoforms. And genome is relatively static, so genetic is usually more static kind of things. But the proteome, it changes constantly in response to tens of thousands of inter and extracellular environmental signals. So it's like basically it change based on your environmental factors. And the proteome varies with health or disease, the nature of each tissue, the stage of cell development and effects of drug treatments. So protein that do the work of the cell, the functional aspects, not genes. And if you look at the drugs, most of the FDA approved targeted therapeutics are directed at protein. So most of, especially in cancers, a lot of drugs, they actually develop based on proteins. So for example, it's this estrogen receptor. So they found this anti-estrogen tamoxifen in 1970s based on this estrogen receptor. And since then, more personalized treatment of patients with breast cancer has been developed. So these are some of the examples. Actually, there are many examples, but just for you to see. So this trastuzumab, initially they developed for general breast cancer. And when first they developed it, the media highlighted it as a kind of a magic bullet for breast cancer. And everyone in UK, they even sell their cars, their houses just to get this drug. But when it's marketed, and then only they realize actually it doesn't respond to all types of breast cancer patients, and it's only responsive to the patients who are having high HER2 protein. So now the companies, they have developed this HERCEP test, which actually can test how much HER protein you have, and then the treatment are given based on the protein. And it's also similar with the cetuximab, which targets on EGFR protein. So the last is the metabolomics and pharmacometabolomics, and this is actually what I'm doing actually in Malaysia. So metabolomics is descriptive study of all the metabolites in a cellular system, and metabolomics is interaction between metabolic property of an organism and the biological and genetic changes. And pharmacometabolics is actually also looking at the metabolites, and then it predicts how the drug is going to respond, whether it's going to cause efficacy or toxicity based on metabolites. It's similar concept with pharmacogenomics, but now it's looking at the metabolites. So the advantages of metabolomics approach is affected by both genes and environment, closer to phenotype, simple and non-invasive, relatively ease of analysis, and potential to identify novel biochemical. So it's actually relatively easy to done this study compared to genomics and proteomics. So these are some of the studies that we have done. So we did one in COPD and one in asthma study, and we're actually comparing those patients who are taking inhaled steroid, and we found actually there are some chemical markers or metabolites which are suppressed by this steroid. So there is a potential that there is a new pathways where actually we can identify which patients are or responsive to steroid. As we know, not all the COPD patients will have response to steroid, and also not all the asthmatics have response to steroid. So based on metabolite level, probably in future we can predict these patients. So metabolomics approach not only may able to diagnose diseases but also phenotype them. And pharmacometabolic approach, which amounts to response targeted drug phenotyping, might provide the basis of a future population screening tool for selecting individuals according to their suitability for treatment with particular drugs, drug classes or drug doses. And even adverse drug reactions could also potentially be avoided, and drugs and dose level could be targeted more effectively. So the challenges of this personalized medicine is, for research, is a limited sample size, because now you are looking at more smaller groups. Instead of looking the whole asthma groups, probably you are just looking at eosinophilic asthmatics or neutrophilic asthmatics. So you are looking at a smaller sample size. So you need to screen larger group of subjects to identify these patients. And to identify these markers to stratify patients is also troublesome. And sometimes the targeted marker may not work. For example, this phase 3 study by Pfizer on bepinizumab for Alzheimer's disease. Initially, they tried to target on carriers of gene APOE4, but it didn't work. And now they did another trial on non-carriers, but it also didn't work. So these are some of the challenges. And lastly is the industries. Okay, so you're looking at pharmaceutical diagnostics and biotechnologies and reduced market size, because you're looking at smaller market size. So conclusion, personalized medicine may lead to a more targeted drug development strategy, and therefore may reduce or prevent drug failure, cause and adverse drug reactions. An increasing number of alliances between diagnostic and pharmaceutical companies, and now we are trying to move from mass market therapies to a more specialized therapies. And genomics, proteomics and metabolomics may also provide a platform for preventative drug development strategy in future. Thank you. Any questions?
I think we don't have much time. If anybody has any questions, you can chat separately.
Okay, I'll be around, so you can just come in.